Hi, good evening everyone. We're going to get started. So I'm Lisa Mongemelli. I'm a faculty member in biology here at Smith. And I'm pleased to introduce Hopi Hoekstra, who is the winner of the 2015 Richard Lounsbery Award. The Lounsbery Award is given by the National Academy of Sciences to American and French scientists in recognition of their extraordinary scientific achievement in biology and medicine. And Dr. Hoekstra is one of only 8 women to have been honored with this award since its inception in 1979. So Dr. Hoekstra is being honored today for her work to understand the molecular basis of how organisms adapt to novel environments. And her innovative work is characterized by its integrative nature, weaving together the disciplines of ecology, evolution, genetics, development, and neurobiology to better understand morphological and behavioral diversity. So she started her career characterizing the genetics of adaptive coat pigmentation in deer mice, and more recently her lab is focused on identifying genes that have contributed to the evolution of burrowing, climbing, and social behaviors in wild peromyscus mice. So her work has a really expansive range, and it creatively addresses fundamental questions in evolutionary biology by understanding behavior at the molecular level, at the ecological level, and everything in between. So as an undergraduate at the University of California at Berkeley, Dr. Hoekstra began as a political science major before being inspired by an undergraduate research experience to make the switch to biology and eventually get her PhD in zoology at the University of Washington. Since joining the Harvard faculty in 2007, she's also been named the Curator of Mammals for Harvard's Museum of Comparative Zoology, a member of the Broad Institute, and a Howard Hughes Medical Institute investigator, one of the very few with interests in evolutionary biology and naturally occurring animal behavior. Her numerous research awards, an impressive list of publications, too many to name here, demonstrate that she's one of our country's best scientific researchers. However, in addition to research, she's really passionate about teaching. She let me know that she teaches the 500-person Introductory Genetics and Genomics course at Harvard, and for that she has also won several awards. So please join me in welcoming Dr. Hoekstra to Smith College for the Lounsbery Lecture.
Thank you, that was very kind. Super. It's a great pleasure to be here. I want to first thank the National Academy and then my local hosts here. It's wonderful to be here at Smith College and meeting old friends again and meeting new ones as well. So what I wanted to do today was to tell you a little bit about work that we've been doing for, I used to say recently, but now this project is extended over almost a decade, but I still think of it as a new project in the lab, where we're trying to identify genes that contribute to naturally occurring behavior. As was mentioned earlier, our lab got started in making these connections between genotype and phenotype with morphological variation. And this image here highlights one of the traits we've been working on, and that is pigmentation. So this is a cute little beach mouse that you can see its coat nicely matches and camouflages in its local habitat, which are on the Gulf Coast Sandbar Islands in Florida. Sort of bolstered by our success in identifying genes contributing to morphological variation, we decided to use the same types of approaches to try to identify genes that contribute to behaviors. And the behavior I'm going to tell you about today is also illustrated by this image, as this little beach mouse is emerging from its freshly excavated burrow. So I wanted to start by recalling something that a now colleague, Ed Wilson, said in the 1970s. And that was the idea that the study of behavior was largely fractionated. And if you wanted to think about this, you could think about a spectrum, and on two ends of the spectrum you may have folks that you would classify as behavioral ecologists, who in general tend to study a great diversity of really charismatic organisms, tended to study them in their natural habitats, and did so to address questions about the ultimate mechanisms driving behavioral variation. So for example, how do these behaviors evolve? Then on the other side of the spectrum, you may classify scientists as neurobiologists and geneticists who had a very different but complementary approach, tended to focus on, for very good practical reasons, a handful of genetic model organisms, tended to study those organisms in nicely controlled laboratory environments, and tended to focus on questions about the proximate mechanisms giving rise to behaviors or behavioral variation. What I think is so exciting is over the last 40 years since Ed made this observation, I think the barriers between these fields have started to dissipate. And why I think this is so exciting is because it affords us an opportunity to, in some sense, take the best of both worlds and ask questions at the intersection of these fields. For example, what is the molecular, the genetic, and neurobiological basis of behaviors that matter for organisms in the wild? And that's the topic I want to touch on today. So I'm going to start by just mentioning a few what I consider fundamental questions. And this is just to give you a sense of the types of questions that we think about. So, for example, what are the relative contributions of genetic and environmental variation to behavioral differences that we observe in nature? So how much is it genes versus the environment? And in cases where genetics matter, what are the precise genetic changes that underlie these fitness-related behavioral differences? And it's not just that we want to know what the genes are, we want to know how they work, how changes in the expression or structure of these genes through neurobiological circuits actually give rise to the behaviors that we're studying. So to answer these questions, we need to start to make connections between genes, neural circuits, and behavior. So some of these questions in particular, the first one is an age-old question. And we may start to think about, well, why don't we have lots of answers or lots of examples of genes that contribute to behaviors? And I would argue that one of the biggest challenges is one that's actually quite simple to at least think about, and that is just simply the idea of how do you quantify behavior. Now, relative to a morphological trait like how tall you are or how light your hair is, actually measuring behavior in a carefully controlled, reliable, replicated, and behaviorally relevant or biologically relevant way is actually quite challenging. And I just wanted to highlight that by putting up what I would argue is among the best studied, most stereotyped behavior, and that is the courtship of Drosophila. So right away you'll be able to tell that I'm not a Drosophila biologist because this is how I'll describe their sort of mating ritual. So they orient to each other, there's some tapping and licking and singing and attempted copulation, and all of these happens before the actual event. Now, if you're going to think about how do you quantify this in a way that's biologically meaningful, not in one individual, but hundreds of individuals, and now you're interested not just in how one species does it, but how they differ between species, I hope you can imagine how complicated this could be. So what's exciting about the field right now, I think, is that we're making a lot of progress in introducing new ways to automate behavioral analysis, which solves at least part of this problem. So I just want to mention briefly one of the approaches that our lab is taking in collaboration with two colleagues at Harvard, Bob Dada and Ryan Adams. And that is what really Bob and Ryan have done is they've come up with a way in which you could take a freely moving mouse in an arena, videotape him moving in 3 dimensions. You can then sort of look at using this sort of heat map approach, the posture of these mice. You can transform these videos into something that looks like this. So if you imagine putting a spline from the nose to the tail of the mouse, which is represented by these images here, and in this heat map where when the mouse, his head is rearing, for example, you can see it's higher in elevation. And what this approach does is essentially allows you to take a freely moving mouse and start to pull out individual repeatable postures over time, and the program will pick this out, and these repeatable units are thought of, we think of them as syllables, and when you string together these syllables, you sort of get the body language of what the mouse is doing. I'm not going to provide you with a lot of details other than to say that this is, I think, an approach that's going to be really exciting in the way we think about, in this case, mouse behavior. But for today's purposes, I introduced this to you because this approach, like many of these new ways to measure animal behavior, really capitalizes on the idea that you take a behavior and you transform it into a morphology. In this case, we're turning this behavior into a series of postures of the mouse. And as I mentioned previously, measuring a morphology is much easier to do than measuring a behavior. But this idea of taking a morphology and relating that to behavior is not a new one. And in fact, in some ways, I think is really nicely illustrated by an idea that was put forth by Richard Dawkins in his book entitled The Extended Phenotype, arguably his sort of second most famous book. And the idea of the extended phenotype is that an animal artifact or an animal architecture, like any other phenotypic product or trait whose variation is influenced by a gene, you can treat that just like a morphological trait. So let's step back. Let me explain what I mean by an extended phenotype for those of you who are not familiar with this. So here are just some of my favorite examples. So here's an Australian bowerbird. This is a male. They're called bowerbirds because males will build these bowers out of materials in their local environments. And what's interesting about these bowers is they're used as a courtship display to attract females. But within a species, often these bowers have a characteristic size and shape and are made of similar materials. This particular species likes to decorate its bower with blue stuff, which females find particularly attractive. Another type of an extended phenotype are nests built by swallows. Again, the size, the position, the location of these nests often is very similar within species, but can vary between species. Another example of an animal artifact or an extended phenotype are the webs built by spiders. And then this last example is one that's slightly more complicated because this is a structure not built by a single individual, but multiple individuals. This is a termite mound. So the idea that Dawkins put forth in this book is that if there's a behavior that's genetically controlled and that behavior results in the production of an artifact, like one of these 4 examples I've shown you here, you can treat this artifact as a trait, just like the length of a femur, for example. And you can subject that to genetic dissection. But as he wrote now some decade ago, he said, 21 years ago, I said that nobody has done a genetic study using animal artifacts as the phenotype. I think it's still true. I think it's still true now. I would admit to disappointment, except that it invites the obvious retort of why don't you get out there and do it yourself then? So Dawkins didn't do it, so we decided to step up to that challenge. And let me first introduce you to the extended phenotype I'm going to tell you about today, and that is burrowing. So burrowing has evolved multiple times in a wide diversity of organisms. Here are just a handful of examples to highlight that diversity. So things like bivalves burrow, ants, gopher tortoises, prairie dogs. Here are more or less realistic representations of those burrows that they produce. Because it's evolved multiple times independently, this suggests that they have some fitness-related consequences, and we can all start to imagine why this can be important. Burrows can be important for predator avoidance, for thermoregulation, for social interactions for those species that meet and mate underground, or for food storage and growth. The other reason we got excited about burrowing is not only its ecological consequences, but because of a beautiful set of experiments done by Carol Lynch, who's a professor at the University of Colorado in Boulder, who, using laboratory mice, showed that at least some components of burrowing seem to be highly heritable. So this suggested that not only is it ecologically relevant, but that genetics may be involved. So it turns out that we decided to focus not on house mice, but instead on a close relative of house mice, and those are mice in the genus Peromyscus, often referred to as deer mice. I'm sure they're incredibly abundant here. I bet if we set out traps, even on campus, you may catch some, if not many. So while maybe to the untrained eye, this looks like a typical mouse, in fact, these guys, these deer mice fall outside of the split between mouse and rat. So if we consider these about 10 million years, these are at least 25 million years diverged. So what that means is they do represent their own evolutionary lineage, but they're closely enough related to our great model organisms that we can borrow a lot of tools from those model organisms, which comes in quite handy. So we study these guys in part because, like laboratory mice, we can bring them into the lab. We can do those controlled behavioral experiments. They breed very well. Like laboratory mice, we're building a number of molecular and genetic tools or borrowing them from these model systems. But unlike laboratory mice, these mice are not commensal, and they're found through a wide range of North Americas. This is just the range of a single species. And in this range, you can find them in almost every habitat type, so the top of the Rockies, the beaches of Florida, the coast of Maine. And because they live in so many habitats, it affords this opportunity for local adaptation. And this is in part what gives rise to a tremendous diversity in traits, including behaviors. So this group has about 55 species in it, depending on who you talk to. But today I'm going to focus on just, at least in the beginning, on one of those species, and that is Peromyscus polyanotus, which is commonly referred to as the old field mouse. It's called the old field mouse because it primarily occupies these overgrown agricultural fields. So I would consider this one of our less glamorous field sites. This is obviously a close-up shot of a burnt peanut field in Alabama. So no offense to anybody from Alabama, but I think we could all agree relative to this field site, which is on the Gulf Coast of Florida, in these wonderful sand dunes off the Gulf Coast, these are the two types of habitats two typical types of habitat that these mice live in. And what's unique about these habitat types is that there's very little vegetation, and they're characterized by this very open habitat. So this particular species of mouse was a favorite not only of me, but of this fellow here. This is Francis Sumner. Francis Sumner I would consider sort of the classic natural historian who spent the bulk of his career driving around the US, catching mice, and describing variation, wanting to understand how variation is generated and maintained within and between populations and species. So he studied, for example, these coat color variants that we have previously worked on, but with his graduate student, shown here, and I love this image because it shows that the convergence between how your advisor and a graduate student dress over time. So this is actually what they would wear in the field. We certainly don't dress this nicely, not even to lecture, right? But they're quite dapper in this attire. But Sumner and his student Carroll did this remarkable natural history study. And what they observed when they went out to the range of this particular mouse was not only did they have these beautiful coat colors, but they built what I consider an extraordinary burrow. And the burrows looked like this. So you could go out into these open fields and you would see a hole and a plume of freshly excavated soil. It was characterized by this long entrance tunnel, sometimes plugged, then a nest chamber, often with nesting material, and then the secondary tunnel that radiates up near the surface, but never penetrates the surface. So next, what I'm going to tell you about is how we trap these mice in the wild, and it's going to highlight two important points about this burrow architecture. Okay, so as I mentioned, we first find the correct habitat type, so we look for open fields, no vegetation. We walk around these habitats, look for holes. Here's this plume of freshly excavated soil, which indicates it's an active burrow. The next thing we do is get a graduate student and have that graduate student, which you can see here, lay down on their belly, face first in that hole. And while it looks like the rest of us are standing around, sort of laughing at him maybe, we're actually cheering him on. In part, initially, as he threads now this tubing down that entrance tunnel till it hits the nest chamber. And then once he hits the nest chamber, he blows into that tubing, and that will sort of get the mice excited. And then here's where our job comes in, because what's going to happen is those mice are going to come popping out that secondary tunnel. We literally jump on them, and our ultimate goal is to catch the mouse or mice that come out of that hole. Okay. So, why am I telling you this, other than it's kind of fun, is that first, that secondary tunnel that I keep referring to is actually used as an escape tunnel. So if something comes in the entrance tunnel, like this tubing, they have an escape door on the way out. Now, importantly, it's an escape door in the sense that, remember I told you that it doesn't penetrate the surface, so we don't know where it is. So you might wonder how even sort of old, slow, chubby professors can jump on these very fast mice. It's because the size and shape and architecture of these burrows are so stereotyped that even though we can't see the opening to that escape tunnel, we can predict where they're going to come out with great accuracy, right? So the two important things are there's an escape tunnel, and the size and shape throughout the range is very stereotyped. So why would they build such a complex architecture? We're starting, actually a team from my lab is going out in the field to directly address this question. They're headed out at the end of the week. But let me just tell you one hand-waving sort of explanation for why they generate such interesting or intricate burrows, and that is to tell you that one of their primary predators are snakes. So it makes sense, perhaps, that if you have things like snakes coming in one direction, that having an escape tunnel out the back is quite useful. Okay, so these burrows have these really intricate shapes, but one of the things we got excited about this particular trait in this particular species is based on work that was done by Wally Dawson, who's often considered the father of Peromyscus biology, because what he did was he took these mice from the field and brought them into the lab and showed that they would recapitulate this burrowing behavior, would build these complex, what I'll call loosely complex burrows in the lab. And using some genetics approach suggested that this was heritable and may be controlled in a very simple way by the underlying genes. So the first thing we wanted to do was replicate the experiments that Wally Dawson did in the 70s. So we built our own boxes. So this is sort of our high-tech facility. So these are big boxes. We have 10 of them, each filled with about 1.5 tons of soil. They're quite big, so they're about 5 feet by 4 feet by 3 feet. We give them a little slope. They tend to like to dig burrows into the side of the slope, and this prevents them from burrowing in the corner. In these, what I'll refer to as our phenodomes. In these phenodomes, we put a single mouse. We put it in there for 2 days, leave it uninterrupted. We try to control food, light, humidity, water, etc., to minimize environmental effects. Then after 2 days, we trap out the mouse, and we use, again, some high-tech technology, which I'll call phenofoam. And essentially, this is the stuff that you buy at Home Depot or Lowe's. If you have a hole in your wall, you squirt it in, it expands to fill that space, and then hardens. So what we can do is do that and make casts of those burrows. So what we're doing is transforming the behavior that mouse performed over 2 nights into a morphological structure that we can measure with a ruler. So next what I want to do is show you just quickly a video of what this looks like. So there's that tube, out popped the mouse. Right before that, you didn't see any escape tunnel. It popped out of that inch or so of soil. And then using the same sort of tubing approach, here Jessie is weaving that tubing down. The phenofoam, well, you'll see it start to come out on this side. And then we leave it in there. It hardens. And then here's where having a lot of very motivated, excited, and strong undergraduates comes in handy because we have to dig these out. This is a relatively small one. And so we can do this. Now, each mouse, as I mentioned, goes in for 2 days, but we replicate their behavior 3 times. So you can imagine now doing hundreds, if not thousands, of mice, each one having 3 replicates. This results in a lot of burrows that look like this. So literally in mountains of data. This is in the early stages. Here's a Harvard undergraduate for scale. And so we have all these sort of morphological representations of behavior. And as I mentioned, you can measure these with a ruler, but you can do more sophisticated things. So working with colleagues Jörg Fritz and Mike Brenner's lab at Harvard, we were able to automate measurement and take all sorts of interesting measurements of these burrow casts. It can identify the nest chamber, the center line, the volume, its curviness, etc. But the 2 things I want to focus on today, we're going to keep it very simple, are 2 traits. And that is the length of the burrow as measured by the sort of center line. So here's a short and a longer one. And then the presence or absence of the escape tunnel. So here's a burrow with an escape tunnel, and here's a burrow without an escape tunnel. Okay, so those are the ones we'll focus on today. So our first question, everything I've told you about so far is about one species, Peromyscus polyanotus. But it turns out there, as I mentioned, lots of Peromyscus species, and we wanted to ask, is this behavior unique to this one species, or is it found throughout the genus? So to do this, we were able to bring mice into the lab from a whole range of species, representative individuals, put them through that same behavioral trial, and ask what kind of burrows do they make. So here I'm just going to show you some simple data related to the total length of the burrow, shown in gray, and the frequency at which an individual burrows over those 3 trials, shown in green. So several things I want you to note from this graph. So here's Polyanotus for reference down at the bottom. And that is that there's a lot of variation among species in the length and frequency of the burrow. Some dig frequently and long burrows. Some don't dig at all. And what's interesting about these that don't dig at all, for example, Peromyscus eremicus is a rock specialist. It turns out they don't dig very much in the wild either. They primarily live in rocky crevices. So we think we're actually capturing some ecologically relevant variation in this laboratory assay. The second thing, which you probably already noted, is that there's a strong correlation between how often they burrow and how long the burrow is they produce when they do burrow. And that's true across these species. But here what I'm just showing you is something about length, but of course one of the most interesting aspects of these burrows is the shape. Well, it turns out that this shape is relatively unique to Polyanotus. So that we have these guys that don't burrow. The ones that build small burrows tend to build sort of this sock-like structure. And this other species that digs a long burrow tends to do so by building long tunnels. Sometimes it bifurcates, but we never see an escape tunnel. Okay, that's unique to this species. And all of this is actually much more interesting when we look at it in a phylogenetic context. So here's a tree of the, showing the relationships among these species based on molecular data. And when we do this, what we see are some things really are not only unique to the species, but are derived. And so that's interesting to me as a geneticist because we're starting to explore the evolution of new or more complex behaviors as opposed to the loss of that behavior. The other thing to note from this phylogeny is that in some cases, even closely related species like these two shown here have very different burrow building behaviors. And what's neat about these two particular species is that they're sister species. They're allopatric in the wild, but if you bring them into the lab and don't give them a choice, they'll happily mate and produce viable and fertile offspring. And that allows us to do genetics. So the first thing we wanted to do was actually ask, in this case, do we have evidence that these differences in burrowing, whether you build a small burrow or this long, complex burrow, is heritable or has a genetic basis? So to address that question, what we did was a simple cross-fostering experiment. So what you could do was take pups of Maniculatus and pups which build small burrows, these Polyanotus that build long burrows. When they're born, you swap them between species, and then ask, does it matter whether the Polyanotus pup was raised by its Polyanotus parents or by Maniculatus parents? So how much are there any postnatal effects based on who you're raised by on your burrow building behavior? And the short answer is there's no measurable effect. Maniculatus build small burrows when they're raised by their own species, as well as when they're raised by the big burrowing species. And the same is true for Polyanotus. Note the difference in the scales between these two. So what this suggested to us is that this is at least consistent with the idea that there's a strong genetic basis. And then we got excited to try to find the genomic regions that were contributing to these differences in these two species. So to do that, we took a forward genetic approach. So what we can do is in the lab, take the big burrowing species and cross it to the little burrowing species and ask what those hybrids built. So remember, these now have a set of chromosomes from the big burrowing parent and a set from the little burrowing parent. And here's the result. So Maniculatus here, we'll just consider tunnel length. Maniculatus build little burrows, Polyanotus long burrows, and F1 hybrids build long burrows just like Polyanotus. Maybe even more striking is that if we look at the frequency of the escape tunnels, Maniculatus never builds escape tunnels, Polyanotus always do, and F1s always do. So what this suggests is that those F1 hybrids make burrows that are perfectly consistent with Polyanotus, suggesting that the gene or genes involved act in a dominant fashion. Okay, this was also important because it allowed us to do the next step of the experiment, and that is we took these F1 hybrids, crossed them back to the little burrowing parents, and asked, Now this next generation, where the genetic material gets all shuffled up, what kinds of burrows do they build? So let's first focus on the length of the burrows, and I just want to show you in some sense the raw data so you get a feel for this. So maniculatus, these are the pure maniculatus, build small burrows. Polynotus, the long burrows. I told you the F1 hybrids build longish burrows as well. But when we look at these back-cross individuals, when we cross these guys with these guys, you see this huge range. So the first thing that tells us as geneticists is that it's not a single Mendelian gene. Okay, perhaps not surprising, and in some ways more interesting. But what was especially exciting is that we still had individuals that build burrows that were just as long as the Polynotus parent. What that tells us is that, okay, it may not be one gene, but it's also not 100 genes. It's somewhere in between. And we can use this distribution to actually guesstimate the number of genes, and we thought there were about 3 to 5 genes. So for us, that was great. It's not sort of a boring single gene, but it's not unmanageable at 100. There were only a handful. Okay, but this, again, is just the length of the burrow. What about the shape of the burrow? So when we started to look at the types of shapes that were produced by these back-cross individuals, we saw individuals that made those small, puny maniculatus burrows, these big Polynotus burrows. Sorry about my ability to draw in PowerPoint, but I hope you get the idea. So that's that big burrow. But what was interesting is that we saw burrows that were very long, but didn't have an escape tunnel, and those that were short that did have an escape tunnel. Okay? And if we simply counted up the number of individuals in that back-cross that built no escape tunnel and those that built an escape tunnel, what we saw was, this is like dream data to a geneticist, right? Never gets this good. It was as close as one-to-one as you possibly could get with real data, right? And what this means is, at least this is consistent with the idea that a single gene controlled the presence or absence of the escape tunnel. Now, I need to sort of tell you two things, for full disclosure, is one, we continue to measure back-cross individuals, and this started to deviate slightly from one-to-one. So, if you have a good result, maybe it's a good time to stop. The second thing is, it's still pretty close to one-to-one. The second thing is that there are other explanations, genetic explanations for this one-to-one ratio. So, for example, it could be a threshold trait, et cetera. So, okay. So now that we sort of had a sense of the number of regions involved, we wanted to know where in the genome these regions were. And so to do this, the next thing we did is take all of those back-cross individuals and genotype them at hundreds of thousands of markers across their genome that told us whether that region came from a big building or small building parent. And then what you can do is you can ask, are there regions of the genome that are predictive of the type of burrow that that individual produces based on its genotype? So here what you're looking at is essentially the association between genotype and phenotype. And here I'm showing you the 4 chromosomes. Each of these little dashes represents a marker. As we moved across these 4, these are the 4 that were significant. And this dotted line represents genome-wide significance. So what you can see here is that as we move along chromosome 1, there's a region of chromosome 1 that's predictive of the length of the burrow. Similarly on chromosome 2 and on chromosome 20. And on this chromosome, however, there's a region that's predictive of whether or not the escape tunnel is presence or absence. So this data is perfectly consistent with what I told you earlier, that we have, one, a handful of genes that seem to be associated with the length of the tunnel, that we have one region that's associated with the presence or absence of the escape tunnel, and thirdly, that we can genetically dissociate the length of the tunnel from the presence or absence of the escape tunnel, because we can get long burrows without and short burrows with. Okay, so this was really exciting, but what the data that convinced me that we may be onto something looks like this. So I'll walk you through this. So what we can do is ask, okay, there's 2 things I need you to remember. One is Polynotus is dominant. Okay? So if we have a Polynotus genotype compared to a pure maniculatus, at this marker shown here, which shows the highest peak on chromosome 1, that mouse, on average, builds a burrow that's 3 centimeters longer. If you take this region and put a single Polynotus allele compared to that has none, these guys build, on average, a burrow that's 3 centimeters longer. And on here, this marker, again, 3 centimeters longer. And if you have a Polyanotus allele at all 3 of these regions, you build a burrow that's about 10 centimeters longer, which is about the difference between the 2 species. There is no reason, a priori or post hoc even, that these are the same in size or that they're perfectly additive. That just turned out to be how it is. This is not sort of Photoshop data. And we still don't really have a good explanation for this. In addition, if we take this region and we put it in a Polyanotus allele, we see an increase in the frequency of building escape tunnels. Okay, so here we got really excited about this. And so we did now a number of other things, is you could take the best burrowers from that generation and cross them to Mammalian, take the best burrowers from that generation and do this over and over. This was the PhD of my graduate student, Hilary Metz. I'm just going to flash through these because depending on what method you use, you can make these peaks look really tiny or you can make them really big. But what she did was took a conservative approach, and here I'm just going to focus on one of those regions that's associated with entrance tunnel length that showed the highest association and is the most repeatable across experiments. And what she did was she identified this region on this particular chromosome and asked, in this region that shows an association, what genes are there? And it turns out that there are 54 genes in that particular region, in the syntentic region in mus. It's actually the same in Paramiscus. And several of them looked interesting based on what we know about their function. So they're expressed in the brain or other pieces of information. For those genes, then, she asked, are they differentially expressed in these 2 species? So this is just a way of us to try to prioritize our list of candidates. And it turns out that there are 3 genes that are differentially expressed in those 2 species. And in particular, this gene that's in the middle of our peak, M5, showed an especially interesting pattern. So let me tell you about that pattern. Okay, so the first thing is when we looked at this expression in whole brains of those 2 species, we saw a difference between Polyanotus and Mammalian. Polyanotus had higher expression than Mammalian. Okay, that's interesting, point 1. The second thing is that if we looked at the expression level in F1 hybrids, shown here in purple, they showed an expression pattern similar to Polyanotus. Remember, they build burrows that are similar to Polyanotus. That was interesting, point 2. And then what we could do was take that F1 hybrid and not ask about its overall expression, but ask about allele-specific expression. Is it the Mammalian or Polyanotus allele that is more highly expressed? And it turns out in these F1 hybrids, it's the Polyanotus big digging allele that's more highly expressed, like shown here. So all these data got us really excited because they showed this beautiful correlative pattern associated with big burrowing. But it turns out this is just correlation. And this got us to thinking, now if we really wanted to functionally test this gene, what would we have to do? And we decided that what we needed to do was actually bite the bullet. This whole time I've been telling you about how we're avoiding measuring the behavior itself. We're measuring its extended phenotype. But we decided we need to measure the behavior. But remember, rodents are nocturnal, and their behavior that we're interested in, they're doing underground. So how do we measure the behavior? Well, for those of you who are old enough, you may remember ant farms. So ant farms were these little things that we got as kids, and they're filled with dirt, and you could watch the ants build their little sort of colonies, and they'd dig out their little burrows. So we made the mouse equivalent of an ant farm. Hope I can get this to, sorry, I hit a corner. There we go. To play. And so what this is this plexiglass on either side, dirt in the middle, a few inches wide. And what we can do then is actually watch the mouse doing its behavior. And you can see all sorts of interesting things, even, I hope, from this video. So when the mouse starts excavating its nest chamber and starts to produce that escape tunnel, it turns over on its back. So instead of digging this way, it gets on its back and digs this way. We could watch these for hours. They're really fun. So I should say we can watch them for hours, but not hundreds of hours. So if you think about it, each mouse, 8 hours of video, we repeat it 3 times. That's a lot of video. There are a lot of Harvard undergraduates, but that's a lot of video. So while cute, we were sort of stuck back in that problem I described before, is how do we actually quantify behavior? Well, it turns out I got very lucky because I had this fantastic postdoc in the lab, Brant Peterson, who automated this. So what you're watching here is this cloud is the mouse moving in space in these ant farms, and what we're recording down here, so you can see it built its burrow, is the amount of excavated soil, so that's the stuff that comes out. And below, we're measuring the amount of visible burrow that this program is picking up. So we can measure all sorts of things, and this is obviously sped up, but we don't actually measure it. The computer does it in this sort of unbiased way. And I should mention we measure these two things, and they're very highly correlated. But we can ask all sorts of things about how the mouse is moving around, when it builds its burrows, et cetera. Okay, so this was really exciting. But what's most striking, I think, is when you compare Polyanotus to a maniculatus. So here's the same setup, and now we're looking at that little digging species. So you can see it's running around. So it starts to build itself a little ramp, and it does these backflips off the side sometimes. It runs around and spends some time over here. If you're getting a little bored, that's the point in some sense, right? It does not really any major digging as the night goes along. So here the lights are going to come on at about 8 hours. And what happens is that this little mouse is running around, and then all of a sudden says, oh my gosh, the lights are going to come on, digs its burrow, and then just sort of sits there until the lights come on. Okay, so even these two videos, I mean, we have now thousands of videos. These are just some stills, but we can start to pick up some patterns, and I bet you've already picked up on some patterns just looking at those two examples. So we started to get interested in the question of what are the mice doing differently that results in these different burrows? And so the first relatively simple question is it a matter of sort of mechanics? Are they digging at different rates? And one of the things we noticed is that, in fact, they do dig at slightly different rates. If they're extending their burrows, Polyanotus does do it a little bit faster, but just a little bit faster. And it only explains about 10% of the difference in the length of the burrows. So one of the things you may have noticed, and maybe not surprising, is that a big difference between the two actually turns out to be the amount of time they spent digging. So Maniculatus quickly dug its burrow and sat in it. Polyanotus spent a significant amount of time digging its burrow, and this really is what explains the main difference in terms of the tunnel length. Okay? This got us excited because we start to think about sort of the underlying neurobiology in the sense that it doesn't seem to be this sort of mechanical difference, like one has better digging equipment in some sense, but it seems to be more associated with what I'll very loosely call a motivational difference for how long they're digging. The other thing that you probably notice from these videos is that when they dig is also very different. So when we put the mice in to when the lights come on, what you can see, where the lights come on in this line here, Polyanotus, almost all of them, you put them into these ant farms, which we often call Brant farms because of this postdoc. In these Brant farms, essentially, Polyanotus immediately start digging their burrow. Like they check things out and then they start digging. Maniculatus, however, wait till just before the lights come on and quickly dig their burrow. So we refer to these as our eager beaver phenotype, and these guys are procrastinators. Okay? And this is, again, sort of reminiscent of this idea that there's a motivational difference in their burrowing behavior. So we started to think about motivational differences, and let me just sort of tell you about the pieces of data and remind you of some of these that got us thinking along this line. So here, remember, there's a difference in the amount of time they spend digging and when in the night or the latency to digging. So Polyanotus seems eager and they keep doing it longer. I showed you this data earlier. That's the length of the burrow and the frequency at which they burrow. Again, Polyanotus is a clear winner. They spend more time to produce these longer burrows, and when they dig more often when they have the opportunity. And then finally, we did this experiment to look at when during ontogeny they built burrows. And this was actually a quite remarkable result. So first, let me tell you that we wean these mice from their moms, the pups from their moms, around 23 days of age. Polyanotus, the first mice to start digging, are 17 days of age. They dig very small burrows, but the complete burrow shape. By contrast, it's not till 10 days later that Maniculatus even starts to build its little teeny puny burrow. Okay? So the Polyanotus is also more motivated to dig burrows in terms of when they start doing it as they grow up. And in fact, we can look at hybrids, and what we've shown is that there's an association between when they start digging as a pup and how long their burrow is as an adult. So precocious diggers tend to build longer burrows, suggesting that they have a shared genetic basis. Okay, so remember I told you about this gene M5, and I told you it had this, it's in our QTL peak, it had this really interesting expression pattern, but I didn't tell you anything about this gene. Okay, so next what I want to do is tell you about this gene, but to emphasize that the data I'm going to show you is largely preliminary. This is one of several candidate genes that we have, but I want to give you a sense of the thinking that's going on into the next steps, and maybe convince you a little bit about why we're excited about this particular gene. Okay, so what do we know about this gene M5 from mouse and human studies? So if we dig into the literature, what we find is that M5 is in fact a muscarinic acetylcholine receptor. It's expressed in dopamine-containing neurons in the basal ganglia of mice, and in particular 2 regions, the VTA, which is associated with reward circuitry, and the substantia nigra, where we can influence motor control, spatial learning, and addiction. Okay, so we could all make up stories about why this might be exciting, but it's kind of interesting. The other thing that was very interesting is that there's a knockout phenotype in mice, and that it's associated with morphine addiction. And in GWAS studies, there's hits in the human genome associated with nicotine addiction. So M5 modulates dopamine release in the reward center of the brain, and this got us thinking about this idea very loosely that maybe Polyanotus is literally addicted to digging burrows. So this is, of course, from mouse. Is it expressed in the same region in Parameciscus? Remember, they're 25 million years diverged. It turns out that we can stain for this in the brain, and in fact, what Hilary Metz, my grad student, found is that it is expressed largely in the same regions. So this is showing you in the VTA, we see expression in dopaminergic neurons, again associated with the reward circuitry. So it's expressed in the right place, but is it expressed in the right time? So here we tried to take advantage of the fact that Polyanotus, as I showed you, starts to dig earlier when Maniculatus isn't digging at all. So what does the expression of this gene look like at these time points? Well, it turns out Polyanotus has much higher expression during these time points compared to Maniculatus. And if we move earlier, it comes on earlier as well. So it's got an interesting phenotype in mouse and humans. It's expressed in an interesting place in the brain. It's expressed at the right time. And we can now start to sort of pharmacologically perturb this gene. So remember, it's an acetylcholine receptor. When we give it an acetylcholine esterase inhibitor, what we see is the length of the burrows get longer with very small sample sizes, I should note. But all of these, I'm not cherry picking sort of the best data from these studies, but these are all sort of pieces of information where we're starting to build our excitement about this particular gene. So here we are asking this question of, is this the right gene? So clearly we have a lot of work to do to actually show that this is a gene that's causally involved in burrowing behavior. Why we're excited about this is because if we can identify a gene, that gives us a handle in which to understand how genetic changes actually, through neural circuits, give rise to differences in behavior. But one of the things I didn't mention to you that I just want to emphasize here is that this gene and this behavior in particular is exciting to us because it doesn't, when I talk about motivation, it's not an overall difference in motivation, it's a burrow-specific motivation. So if we give these mice any other tasks to perform, motor-related tasks, like time spent on a wheel or distance run on a wheel, if anything, it's Maniculatus that's more active. So it's really that, is this the right gene? How does it work? And in particular, how does it work to change your motivation for one particular behavior, but not others? We don't have the answers to this, but I think this is why we're so excited about this particular project. So let me just tell you about sort of the next steps very quickly. So we're continuing to use our genetic mapping approach to narrow in on these regions to get a better handle of our candidate genes. We're also mapping differences in behavior, not just the extended phenotype. We hope that these results are complementary. We're also using comparisons of gene expression in the brain. We can look at expression between species, in particular brain regions like the VTA, across ontogeny, before and after digging, or allele-specific expression differences. We're also starting to look at the neurobiology of burrowing. So we can use viral injections, and now with the advent of CRISPR-Cas, we can start to manipulate particular genes in particular regions, even in non-model organisms. And then finally, we're also doing some experimental manipulation. So here's one example. If you give a mouse a burrow that's already half its length, well, that mouse that usually produces a 20-centimeter burrow, and you give it a 10-centimeter burrow, will its burrow be 30 centimeters, or will it be 20 centimeters? So in other words, are they measuring how much they build or the final product? And we hope that these types of studies will help illuminate the neurobiological mechanisms. Finally, I want to end by telling you that these species, while producing these, I think, remarkable charismatic differences in burrowing behavior, also differ in a number of other behaviors. So, for example, old field mice, which remember live in these open old field habitats, when you put them in an elevated plus maze, which is a measure of sort of anxiety-related behaviors, or how anxious a mouse is when it's exposed. So this is a maze in which it's elevated off the ground. 2 of the arms are closed, 2 of them are open. What you can see is Polyanotus is quite comfortable in these open arms compared to Maniculatus, consistent with the habitat differences in which they evolved. There's also big differences in their mating systems. So Polyanotus is one of those rare examples in mammals in which the mice are both genetically and socially monogamous, whereas Maniculatus is a more typical mammal, mating with whoever they run into. And these differences in mating system have big implications for parental behavior, and in particular, paternal behavior. So here's an example of time spent huddling. I don't know if these images are, if you can see them very well, but here Polyanotus is huddling, Maniculatus huddles much less. So the mothers are both pretty good, right? Maternal care is universal in mammals, but we see a big difference in fathers. So in Maniculatus, our promiscuous species, moms huddle the pups, dads don't do so much. But in the monogamous species, the moms are really good huddlers, and the dads are equally good. And this, of course, then has big implications for reproduction, because if promiscuous species are mating with lots of individuals, there's different levels of sexual selection, which transfers into different levels of reproductive traits like sperm morphology, where the promiscuous, competitive Maniculatus sperm have bigger midpieces, which make the sperm swim faster, and this has big effects on their reproductive fitness. So here I'm just hoping to give you a glance of all the types of traits that we can start to understand in this particular species pair, with our ultimate goal being trying to identify particular genes that contribute to these differences. Now, for many groups, finding a gene represents an endpoint. For us, finding a gene represents a handle at which we can then understand mechanism. And we're interested in not only the molecular mechanisms, where are these genes expressed, what are they doing in neural circuits, or how do they affect neural circuits, and how do they act to have specific behavioral effects. But we're also interested in evolutionary mechanisms. Where and when have these alleles arisen? Is there evidence of selection in the genome? Does the same gene contribute to convergent phenotype? And what I've hoped to have done today is to convince you that by combining the best of behavioral ecology and neurobiology and genetics, we're starting to be able to make these connections, or there's hope to make these connections between genes, neural circuits, and behaviors, in particular for behaviors that matter in the wild. So let me thank the people who did the work. So Jesse Weber, who's at UT Austin, was the brave graduate student who took on this project. He was soon joined by Brant Peterson, who did all the work in ant farms I told you about. The project was then taken over by Hilary Metz, who did sort of the hard genetic work, and Nicole Bedford has been working on this in the ant farms and then headed out to the field at the end of the week. With that, let me thank our whole lab group. Literally everybody in our lab has gotten their hands dirty. a huge group of undergraduates and our funding sources. And with that, I'm happy to take any questions. Yeah, so the question is about what do we know about the genetics of the escape tunnel? So everything I've told you about today, M5 seems to be associated with entrance tunnel length, and that's for practical reasons, in part because that shows the largest association, very repeatable. We haven't followed up so much on the presence or absence of escape tunnel. That's certainly on our list of things to do. There's two challenges to that. And one is that measuring the escape tunnel can be a little bit more tricky, because if the burrow is small, sometimes knowing whether there's an escape tunnel there or not is hard, so it's a little bit noisier. The second thing is that in terms of the genetics, the region that's implicated is at the end of the chromosome, and that ends up being harder to map genetically. But as we're building the number of back-cross mice we've generated and so forth, we're hoping that we can hone in on that. We still haven't ruled out the possibility that there are multiple regions involved that have this, you need 3 of the regions. Let's say there are 5 regions associated with the escape tunnel, and if you have 3 of the polyonotus alleles, then you build one, which then would be more complicated genetically, but I think also interesting. Yeah? So the question is, how do we estimate in the field, you mean, where the escape tunnel comes out? So the way to think about this is that the size, the length, and the angle they produce, that sort of where that nest chamber is so predictable that if you're standing there and you can see where the burrow goes in, you can essentially say, here's where the escape tunnel is going to be. Now that's not quite right in the field, because sometimes there's a root or a rock or something, but so much so that I can't tell you, first, for anybody who's worked with mice, they're fast. Let me tell you, these mice are much faster, right? Like laboratory mice, they're pretty fast, but these guys, the fact that we can catch not just one, but sometimes multiple mice that come out I think illustrates that we have some clue where that escape tunnel is, and a pretty good clue. Yes? Okay, so the question was, and I think this is a very fair, very observant point, that I said that they're sort of motivated, and again, I just note the quotes, it's a little bit of a loaded term, to dig burrows, but not other behaviors. And maybe the better way to phrase that, or more precise way to phrase that, is that there's not an overall difference in activity level. So it's not just that polyonotus is going to dig, and it's going to run, and it's going to move, and they're just more active overall. I think to address the question of if they're more motivated overall, we need to use another assay that specifically tests motivation, which we are doing but don't yet have the results to. So I think that's a really good observant point. Yes? Mm-hmm. Okay, so the question is about, are the predator differences between these two species, and is that correlated with when they dig their burrows? So the first thing I'll say is that the differences in predators is just a fundamentally difficult question to answer. We know something about polyonotus because they're range is fairly small and they're in a very specific habitat type. But Maniculatus is the species that's sort of everywhere else in the US besides the southeast, so their predators are going to vary tremendously. So we have 1 point of each of those, so it's a little bit hard to know, but I think it's fair to say that there are sort of the major predator types, like hawks and snakes and things like that, are present in both. What I think is another explanation for why their digging time is different, and again, we don't have data to support this, but I think this is a plausible hypothesis or story, is that old field mice, right, live in these very open habitats. When they go out of their burrows, they're exposed to predators immediately. There's sort of nowhere to hide but their burrows. Compared to Maniculatus, which are in prairies, dense prairies, or fields in general, where they can run up a tree or a rock or so forth. Maniculatus, so there's sort of like, I got to dig a burrow if I'm exposed because that's my only hope. The other thing may be a trade-off with finding mates. Remember, Maniculatus is very promiscuous and may see that as a higher priority. It spends more time, during the day during their day or night, finding a mate, and then it's like, oh no, I got to build a burrow, but that's low priority. Whereas Polyanotus, building a burrow is important, but they're monogamous. They have their partner. They spend less time needing to find those mates. I don't know what the answer is, and this is a hard question to get at, but I think it's really fun to think about. That is a good question. So what I can tell you, I can't answer that question about do the mice that build these big burrows and are in there together and are monogamous, in the context of burrows, do they have different levels of oxytocin? What I can tell you about is that we have looked at expression differences in whole brain at a number of different genes. There are differences in oxytocin between the two. There are also differences in vasopressin. We're doing this but you take two species and that could be biologically important, or it just could be that they're different species. So one of my postdocs, Andres Bendeski, has been mapping changes in paternal behavior, which I sort of hinted at here, and has implicated potentially a role for vasopressin in male paternal behavior, which is quite interesting, and we're starting to follow that up now in a functional sense, and the results I think are quite interesting. We don't see any mapping of parental behavior to the oxytocin or oxytocin receptor or vasopressin receptor in our mapping experiments. But we didn't look at huddling per se, we were looking at parental behavior in particular. It's a great question. There was somebody here who I've been neglecting, yes. Yep. So do we have enough molecular data to look at genome-wide levels of selection? So I can tell you that hopefully next Wednesday we will have a beautiful, complete assembly of the Peromyscus polyanotus genome. So that's been a long haul. So that's been done at the Broad Institute. Doing resequencing experiments, I think we could do. The one caveat is that these scans of selection across the genome are very good at picking up selection on the genome at a particular time scale. So it has to be fairly recent. We know that knowing when these two species diverge, some people say 1 million years, some people say 20 million years, but we don't, independent of when they diverged in some sense, we don't know where along the Polyanotus lineage, assuming it is really derived and evolved in that lineage, this complex burrowing behavior evolved. It could have been 20,000 years ago, and we'd be in great shape, but it could be 20 million years ago, and then those approaches won't work. But of course, we'll try. Are there multiple alleles of M5? So what we know about M5 is that there are differences in, so you're talking about mouse populations or human populations? Okay. Mouse populations, we know that if you compare the two species, there are amino acid differences in this G protein coupled receptor. We know a lot about GPCR structure and function. Some of those are predicted to have an effect on both structure and function. We've done some pharmacological assays, and we don't see any measurable differences, but of course that's a result. It could be, if we did it in another condition or another cell line, we could. So we know that there are differences between the two species. We have done less looking within species, but there's nothing that sort of jumped out within species yet, but we haven't done a really good survey yet. Okay, so is it expression or amino acid change? We have no idea, and you could have both. And so we put the structure stuff on the side for a little bit before sort of building more evidence that M5 was really important. And I think if we get to the point where we've convinced ourselves that this is a really, really good candidate, and we start doing, for example, manipulate this through CRISPR or something, we can look at both expression levels as well as trying to make some of these amino acid changes. At this point, we see some evidence of both, but neither one we've nailed down yet. That's a good question. Oh. Okay, this is a great question. So how often they actually do this in the wild? So the first thing I can tell you with Maniculatus is that their burrows are really hard to find in the wild. They're small, and they live in forests, and it's very different than the old field, right? Like it's open area, you can see the hole, the plume. So we know very little about Maniculatus, and my guess is that the burrows of Maniculatus may actually look different depending on where they are in the range, although we don't know. For Polyanotus, what I can tell you is two sort of anecdotes. One, if we go out in the field and we see a burrow and you see the entrance tunnel and you see the escape tunnel, like it's blown out, we've never found a mouse in a burrow like that. Which suggests, and that's probably a good idea, your secret escape hatch is blown, now you can have predators coming in two ways, which suggests is that they probably have another burrow. I don't know, it seems like a smart thing to do, that if you're gonna ruin your one house, that you have another house you could go to. So what this field team that's going out on Friday to Florida is to ask exactly those types of questions. So we're labeling mice with little RFID tags. We have little antenna by burrows. We're doing mark recapture studies. All of those types of things to address these questions, because even though I sort of told you this is ecologically relevant, we haven't done the experiments to really show that. And there's a bunch of missing, I think, natural history data about how they use these burrows. Can I tell you one thing really quick that's related? We did this experiment. So everything I told you about was the single mouse digging a burrow. But I also told you that lots of mice come out of these burrows, right? So what we did is we took two mice and we put them in a box and asked, what do? Do they each build their own burrow? They build one burrow, and it's longer. So I think things in the field are going to be even more interesting to ask. Do they share burrows? Was it always with their partner? Et cetera, et cetera. So it's a little bit more complicated than a single individual.